Hello Year 12, welcome to another flipped video. Alright, so this is a relatively big one, and I think will warrant one or two views, mainly because we're dealing with plants, and plants have systems that we may not fully be familiar with, and because of that, it might just need revisiting. So, let's get to our dot point. So, by unpacking this dot point, we are looking at the verb, and our current verb is describe. So we are looking at features. So we need to kind of get into the mode of kind of looking for things that stand out that might be relevant in the content that we're looking at. And the content that we're looking at is current theories about processes responsible for the movement of materials. And we're looking at that content through the context of plants in xylem and phloem tissue. Now, xylem and phloem tissue is taught every single year from Year 7. But we constantly find that even in Year 11 and Year 12, for students who pick biology, that they are still not familiar with it. So this video may be a little bit longer than what you were expecting, but we're going to kind of really give you a good grounding in what's going on in plants. Alright. So the first thing is identifying that there is a bit of an issue in plants. They're a bit different to animals in the sense that they have they are similar in the sense that they have cells all over their bodies. It's a common feature of multicellular organisms in the sense that some cells require, all cells require materials, but they have a problem. The problem is that roots can obtain water but not sugar, and leaves can produce sugar but can't get water from the air. So there needs to be some sort of mechanism of exchange, and so this is what we're looking at today. To kind of, get you a little bit familiar with what you might be seeing in textbooks, in exam questions, and maybe even in other stimulus that you might be coming across, we're looking at this idea that this transport system is done with vascular tissue. Done's not the right word. That is supported by vascular tissue. So over here, you'll see we've got these little, in the stem, we've got these little nodes, I guess you call it, or bundles. These are vascular bundles, and within these vascular bundles there are different types of tissue, and the ones that we want to really pay attention to are the red circles here. They are xylem, and these ones on the outer here are phloem, these smaller ones here. So you've got xylem and phloem, and in here in the root you can see you've got the red large circles. They carry lots of water, and then here we have our xylem. So these little nooks of more compact cells here. Interesting to note though, in the root cells, these purple little splotches inside of our other root cells here, these are starch molecules, or starch granules, which actually is evidence to say that they do in fact receive sugary substances from the leaves. So that's pretty cool, showing up here. Over here in our leaf, we have our red stained xylem, and then we have this little layer of little cells here in our vascular bundle of phloem. Below we have our lower dermal cells, and we have our upper dermal cells. So the sunlight is hitting this layer here, and you can see our palisade cells here with lots of chloroplasts in there. It's not really relevant to our conversation, but just to kind of orientate you with the leaf here. And below we have, we can see this is the underside of our cell. We have some guard cells here. So this is the stomata there, and I reckon I can see another one over there. So we're looking at the bottom of the leaf and the top of the leaf there. Alright. So, one of the things that I really wanted for you guys to have was a big picture overview of transport in plants. So I've provided this to you guys, and it gives me a way of ensuring that we're talking about the same thing. So I'm going to start with talking about the movement of water. And immediately if I say water, you're going to be looking at the H2O, and we're going to be looking at this blue stream. And you can see that it goes from the roots to the leaves. And that's one way you can think about it. The other way you can think about it is that the leaves lose water and things are pulled up. So, you know, keep an open mind about it. It works both ways, but it certainly doesn't. I mean, even though the arrows start at the roots, it can be caused by something going on in the leaves and the loss of water from there. All right, we have a green arrow coming down here. This is when I'm going to be talking about our sugar. The movement of sugar. Sugar is produced by photosynthesis, which requires light, and can travel anywhere that the plant desires that sugar. So that's it does go down, but it can go up. It can go to other leaves. It's pretty cool like that. We have minerals and some gases. Now these aren't super relevant to our conversation today, mainly because we are only talking about xylem and phloem tissue. So I wanted you to know that there was processes here, but we are only looking at the blue and the green processes here. Okay, let's get stuck into it. So the first one is we are looking at water. It's the transport of water. We're looking at a process called the transpiration stream, or transpirational stream, depending on where you might see it. And basically it has three areas that we need to look at. We have water that evaporates from the leaves, so we know that the top of the leaves gets a bit of sun, and our stomates open and close to obtain the gases, and when they open and close they can lose and will lose water from evaporation. So that means that there is a lack of water. There is a negative balance of water in the leaves, and that comes into play a little bit later. We have another factor of capillary cohesive forces in the xylem tissues. So this is called capillary. You might hear it called capillarity. You might hear it called capillary action. It depends on how it's kind of labelled, but basically if we're talking about capillary, we're talking about the movement of materials in the stem going up. It's generally the upward movement here. And then we have osmotic differences in the root, where root hairs pull water into the roots. Okay, so that's kind of three areas here that are working. And you'll notice that this is the label here is passive, so there's no energy required for this. So let's have a look at capillary cohesive forces, now capillarity action. So basically water has a couple of properties. First property is adhesion. It likes surfaces, and we know this. You know, you'll have a shower and you'll flick the water onto the shower curtain or the shower window, and it'll stick to it. We know it doesn't just immediately like drop off the wall, it will stick to it. We know water has sticking properties. But water also likes itself. So water will, the reason why kind of water falls in droplets from the sky is that those water molecules are bonding with each other, and so they're not just going to kind of loosely separate, they stick with each other. And this comes into play when we put these two features come into play when we put the water into tubes. So water will stick to the edge of the tube, and it will also stick to each other, and so you get what we call a meniscus. And the thinner the tube, the higher the water will go. We can demonstrate this with glass, although just about everyone will have seen this when they stick a straw into a glass of water or Coke or milk or whatever. You will see that the water will rise higher than the water inside the straw will rise higher than the water in the cup. This obviously, just by this diagram actually probably explains it a little bit better. The narrower the tube, the higher the water will go. So this is actually pushing the water up the hill. Now, if you were a water molecule leaving the leaf and through evaporation, you would be pulling on other water molecules behind you. And so that process of evaporation and cohesion helps pull water molecules up the stem. Hopefully that makes a bit of sense. This happens inside of xylem tissue, and this is where we're going to explicitly address this idea of what xylem is. So xylem is formed from dead cells that have had their end of their cell walls removed, and so they're kind of these long dead tissue, and they're just essentially little tiny tubes. And they're strengthened with a substance called lignin. And lignin is probably you'd probably most be familiar with it with the association of strength of celery stems. You know that celery stems are a strong stem, and it's because of this lignin that is occurring there. So there's no ends. It can transport minerals, water and minerals, and it only goes in one direction, and that is up. Alright, so that's xylem. So we're going to talk about water in the roots. Water in the roots. So we've got, if there is lots of water in the soil and there's not much water in the plant, the shear osmotic balance indifference will cause water to move into the root cells. But if that isn't the natural state of things, if that's not, if there's not much water in the soil, the plant can actually force water to move in by actively pulling salts through the sodium-potassium pumps into the cell, creating an osmotic balance, and this causes water to passively move into the cell. So despite the process of pulling salts into the cell being active, water moves passively. So this whole process of moving water doesn't require any energy whatsoever. Evaporation is caused. The loss of water from the leaves is caused by evaporation. That isn't a drain of energy off the plant. You have capillarity. That's just the physical forces of water working within a tube, and then you have this osmotic balance being established here in the root of a tree or root of a plant, not requiring any energy either. So I've added this video back, this slide back in because I think it's kind of good to kind of recap where we were, and it's kind of should get your head back in the game here. We have the blue stream moving up into the leaves through the processes we've just discovered. Now we're going to talk about our movement of sugar throughout the plant. Sugar, and we basically mean sap. Sap can have other salts and maybe other proteins and even hormones in it, but it's generally it's mainly sucrose. In the leaves, photosynthesis produces glucose, and this is almost immediately converted through a metabolic reaction into sucrose, so that it can be stored more efficiently. So let's have a look. Alright, the theory for moving sugars around a plant is called source to sink, or bulk movement. Some American sources, some American sites, institutions, whatever, call it bulk movement, so you might want to just be familiar with that one as well. But we're going to talk about some basics. So phloem sap is, sorry, I'm going to have to cough. phloem sap is an aqueous solution that is mostly sucrose, and it travels from a sugar source to a sugar sink. Tongue twister. Alright, so a sugar source is any site of photosynthesis, such as mature leaves or even young leaves. But basically, if you are photosynthesizing, you are a sugar source. A sugar sink is an organ that requires sugar. So that could be a tuber, or a bulb, or a fruit, or a bud off the edge of like a point of growth, like a little bud off the edge of a stem or a branch. So those sinks can be anywhere on the plant, and the source is anywhere that photosynthesis can occur. So this is pretty cool. It's already kind of suggesting that because the location can be all over the plant, that the movement is any which way that the plant requires. Alright, so this is kind of difficult to understand if you're not prepared to look back at those slides that showed how close xylem and phloem were together. Basically, we'll go through it and we'll see how we go. So, we have a source cell, so this will be a cell or some tissue near a photosynthetic site, and it actively pumps the sugar produced from photosynthesis into the phloem tissue, the sieve tube. So there's these little green blobs here. And you'll notice that where the source of sugar is, there are a lot more green dots. Now, because there are a lot more green dots here of sugar, the osmotic balance is water will want to move from a low solute concentration into a high solute concentration, and so you see this water movement over here. Once you get a lot of water, you have an enormous amount of pressure, and this pressure will only go down to areas of lower pressure, and so you start getting this movement. You get the water pulling and pushing the sap down the phloem tube. The pressure is relieved by sink cells, so cells that are requiring the sugar. They actively pull the sugar molecules into the cell so that they can use them or that they can store them. And so by pulling the water molecules out, they reduce the solute concentration, and water will squeeze back over, or diffuse, or via osmosis into the xylem tube, and the process can start all over again. What this diagram doesn't really show, and it kind of does, is that these cells here and here are also considered companion cells. So xylem is pretty much standalone, but phloem tissue has the tube itself, but also cells that are attached to each part of the phloem tube that can actively regulate this pressure all over the plant. And so you've got to think of these not only as a source cell and as a sink cell, but as the means of regulating that pressure. I think that's a pretty important concept that this one particular diagram didn't show. I'm sure we'll come across it in other materials that we look at in class. Okay. Alright, so in order to get our head around all of that information that you've just received, I've tried to table it. Now, I've received feedback from people saying that they quite like the tables, because it's a kind of a nice summary. But if these aren't working for you, then we'll kind of we'll have different things we can use in class to help you move along with this. But we are looking at a table of features, I guess, descriptions. So we've got the name of our current theory for the movement of water, and that's the transpirational stream. we've got the name of our current theory for the movement of sap and other materials, and that is source to sink. The tissue used for transpirational stream is xylem. It's passive. The movement is upwards only from the roots to the leaves, and the mechanics involved, now these are the kind of like the features that you'll need to include, is basically you get transpiration in leaves, the loss of water through stomates pulling on water molecules below it, capillarity in the xylem stems. You have cohesion and adhesion factors at play there, and then you have the osmotic balance in the roots. Over here though, in for source to sink, we have phloem tissue used. We have the type of transport, which is active, and which means it requires energy. Cells are actually doing something. In this case, they are regulating pressure. And the direction of transport is any direction necessary because of that active process allowing that regulation. And the mechanics of that is the pressure flow. So it's the companion cells can actively pull sucrose in and out of phloem tubes to regulate pressure and therefore direction of flow. That was a big video, boys and girls, and I hope that it wasn't too overwhelming. You can't avoid plants in the biology course. We do a lot on other animals, but the course always stings you with a bit of plant stuff, pretty much in every topic. So look, it's just how it is. So if that was familiar to you, that's great. But if it's not, then let's work on this in class. Again, it's really about nailing the dot point in terms of the content and the context. So we're looking at the in xylem and phloem tubes tissue. What is going on? Well, here we have movement of water and other materials, and our features are here. Hopefully that's not too hard to explain. There probably were a couple of diagrams that might help, namely that overview diagram, but that's up to you whether you're a visual learner or you like the segmented table versions. Okay, folks, I'll leave you to it.